Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CSIR Net JRF Life Science question paper conducted in the year March 2025. This is part B section that I am discussing. This is of the morning shift paper or the shift 1. If you are new to my channel, please do like, share and subscribe. So coming to the first question. Which of the following modifications in their native system does not lead to translation inhibition? Not. That means translation inhibition is not caused. Okay. So we have to see. So nucleotide addition resulting in incorporation of a stem loop structure in the mRNA upstream of the Shine-Dalgarno sequence. Now what is this Shine-Dalgarno sequence? You all know, but still I am just brushing up your memory. So it is a sequence that will help in the binding of the ribosome to the mRNA. So the ribosome has to be bound for translation initiation. Now Shine-Dalgarno sequence, it has got an anti-Shine-Dalgarno sequence at the 3' end of the 16S rRNA in the small subunit of the ribosome. Okay, so this is the Shine-Dalgarno sequence present in the mRNA will bind to this anti-Shine-Dalgarno sequence in the 16S rRNA through complementary base pairing. And this is the Shine-Dalgarno sequence. It is found upstream of the start codon. Okay. And this will help in the binding of the small subunit of the ribosome to your mRNA. And this is found in prokaryotes mostly. So now what they have told us, which does not lead to translation inhibition. It will not lead to translation inhibition. Which out of these? So nucleotide addition resulting in the incorporation of a stem loop structure in the mRNA upstream of the Shine-Dalgarno sequence. That is, your Shine-Dalgarno sequence is here in this region. This is upstream region of the Shine-Dalgarno sequence, and you add nucleotides here such that here there is a formation of a stem loop structure. Now will that hinder with the translation? No. Why? Because your Shine-Dalgarno sequence is intact there. It is not harmed. It is not involved in stem loop structure formation. It can form complementary base pairing with the anti-Shine-Dalgarno sequence in the 16S rRNA and result in translation. Okay. So that will be your answer, but I will discuss the others also. So it doesn't lead to translation inhibition. Okay, so that will be your answer. That is what they have asked in the question. Then next option, if you see what they have told, nucleotide addition resulting in Shine-Dalgarno sequence being part of the stem loop structure in mRNA. Now you add nucleotides into the Shine-Dalgarno sequence, and of course, in such a way that it will result in a stem loop structure. Then Shine-Dalgarno sequence cannot bind to the 16S rRNA and it cannot recruit the ribosomes to mRNA. So translation will be inhibited. So that cannot be the answer. Then next one, okay, I will discuss in the next slide. 1 minute. Expression of an EIF2 mutant that mimics its phosphorylated state. Okay. Now EIF2, they have jumped from the prokaryotic system to the eukaryotic system. So EIF2, it is involved in the initiation of translation in eukaryotes. Now what is this EIF2 doing in eukaryotes? We will see. EIF2 is bound to GTP, then it will bind MET-tRNA I complex, and this ternary complex along with other initiation factors will result in the formation of 43S pre-initiation complex. This is it. Then this along with the RNA and other initiation factors will form the 48S pre-initiation complex. This is it. And once this scans the mRNA for the AUG start codon, then it will result in the binding of the larger subunit of the ribosome to the mRNA. So here is your AUG codon that is recognized by this pre-initiation complex upon binding to mRNA, and it will result in the binding of, once that AUG is recognized, it will result in the binding of the 60S subunit, and there is a complete 80S initiation complex that is formed. Now what will happen to these initiation factors which are bound to the pre-initiation complex? They will move out. Okay, they will be removed from the binding to the small subunit. Now out of these initiation factors, I will be discussing only the EIF2 one. Now EIF2, it will be removed by hydrolysis because EIF2 was bound to GTP. Now it will be hydrolyzed from GTP to the GDP form, that is by the action of GTPase activating protein. So it is bound to GDP. Now it cannot form complex with met tRNAi. Okay. Now what has to be done? It has to be converted from the GDP bound form to the GTP form, and this is done by guanine nucleotide exchange factors, and this will result in EIF2 getting bound to GTP, and it can help in the initiation of translation. Okay. Now in the question they have asked expression of an EIF2 mutant that mimics its phosphorylated state. So it is a mutant of EIF2 and it is mimicking the phosphorylated state, that is it is always bound to GTP. So it will bound to GTP one round. Okay, it can help in translation, but it will not be converted back to GTP once it has formed EIF2 GDP complex. So it will not be converted back to GTP. Okay. So it will result in translation inhibition. Then a mutation that lead to decrease in the processivity of capping enzymes that leave numerous mRNA devoid of cap structure. Now you know that cap structure, it is essential for ribosome recognition and initiation of translation. So there is a decrease in the processivity of capping enzyme because there is a mutation in the capping enzyme. So this will leave so many mRNAs which are uncapped, and uncapped mRNAs, they are not likely to be recognized by the ribosome. So it will also result in translation inhibition. So your answer is option 1. Okay, next question. The CAM kinase 2 act as a molecular memory device as well as a frequency decoder of CA2 plus oscillations. Which one of the following statement regarding CAM kinase 2 is incorrect? So they have asked for the incorrect statement. Now first statement, okay. Now what is this CAM kinase 2? It is a CA2 plus calmodulin dependent protein kinase, and it is involved in various cellular process in the brain, and they have told it is involved in the molecular memory device in the brain. And CA2 CAM kinase 2, it is activated by CA2 plus and calmodulin, and once activated it will autophosphorylate itself, and it can And this autophosphorylation, it takes place at threonine 286 position, and it can remain active even after the Ca2 plus and calmodulin have decreased in the concentration. Okay, so so we will see the statements. CAM kinase 2 activity increases as a function of Ca2 plus pulse increase. That statement is correct. Then CAM kinase 2 requires Ca2 plus and calmodulin to remain in an active state. No. Even if the Ca2 plus and calmodulin levels decreases, CAM kinase 2 can remain in the active state. So that is the incorrect statement. A brain-specific CAM kinase 2 knockout mouse will have difficulty in remembering where things are. So that's correct. CAM kinase 2, they have told in the question, it is a molecular memory device. So when CAM kinase 2 is knocked out, means the mouse doesn't have that CAM kinase 2 in them. So they will have difficulty with regards to their memory. So this will lead to difficulty in remembering where things are. Then molecular memory function is lost in an autophosphorylation defective CAM kinase 2. Of course, because when Ca2 plus calmodulin is present, the CAM kinase 2 gets activated, and upon activation it will autophosphorylate itself. When there is a loss in the autophosphorylation activity of a CAM kinase 2, the CAM kinase 2 cannot remain active. It will not be activated. It will not be autophosphorylated. So what will happen? They have told in the question, CAM kinase 2 act as molecular memory device. So naturally molecular memory function will be lost. So that statement also is correct. Now next one. Which of the following statement regarding cytoskeleton proteins is incorrect? Okay. Now they have asked for the incorrect statement. Actin and tubulin proteins appear to have arisen from single copy genes present before multicellular eukaryotes diverged. That is correct statement. That is regarding the evolutionary origin of actin and tubulin. Now tubulin specifically binds ATP while actin preferentially binds GTP. That is the incorrect statement. Actin binds ATP and tubulin binds GTP. So remember, actin A, ATP binding. Tubulin T, GTP binding. Okay. So answer is option 2. Then FTS is that a tubulin relative forms a filamentous ring needed to effect cell division in certain prokaryotic species. Yes, that is also correct. FTS is involved in cell division in prokaryotes. Then MERB, an actin relative, form filaments required for the cell shape in certain prokaryotic species. That is also correct. Then next one. Glioblastomas, oligodendrogliomas and astrocytomas harbor mutations in isocitrate dehydrogenase. Okay. The IDH mutation found in these cancers cause the enzyme to convert isocitrate into the oncometabolite. Oncometabolite because it disrupts the normal function or the normal cellular process. That is what oncometabolite does, which accumulates in cancer cells. This oncometabolite works by inhibiting several enzymes that require DASH for their function. Now these are certain cancers, and in these cancers the common thing is that they have mutations in their isocitrate dehydrogenase gene. Now what does this isocitrate dehydrogenase gene does in the wild type or the non-mutant persons? So you know that isocitrate dehydrogenase, it is involved in the citric acid cycle, and it helps in the conversion of isocitrate into alpha ketoglutarate. That is what it does. So, and this alpha ketoglutarate, it will be used by various other enzymes for various functions. Okay. Now, what will happen in these cancer cells is that they will have this isocitrate dehydrogenase gene mutation. So, the isocitrate dehydrogenase that is produced from these mutated genes, it will have different catalytic activity. And because of this catalytic activity, it will convert isocitrate to 2-hydroxyglutarate or 2HG, and this is an oncometabolite. So your first blank, this fill in the blank, this should be 2-hydroxyglutarate. So that makes option 2 as not correct, whereas in others it is 2-hydroxyglutarate. Okay. And this 2-hydroxyglutarate, it is an oncometabolite, and it accumulates in these cancer cells, and it will inhibit those enzymes that require alpha ketoglutarate for their function. Okay, now alpha ketoglutarate is what is present in the wild type or the non-mutant, and I told you it is required by several enzymes for their function. But this oncometabolite 2-hydroxyglutarate, it will inhibit the various enzymes that require alpha ketoglutarate for their function, and this can cause further complications in your cancer. So the answer here is 2-hydroxyglutarate and alpha ketoglutarate. So which accumulate this oncometabolite works by inhibiting several enzymes that require alpha ketoglutarate for their function. Okay, so answer is option 4. Then, which of the following statement regarding mutations in the mitochondria during aging, so they have asked regarding aging, is incorrect? Mutations in mitochondrial genome leads to defects in energy production. That is correct. Because mitochondrial is the site of ATP synthesis through oxidative phosphorylation. So mutations in the mitochondrial genome can lead to defects in energy production. That is correct. Then mutation rate in mitochondria is much higher than that in the nucleus. Yes. There is limited DNA repair mechanisms in mitochondria when compared to that in the nucleus. And mitochondria also result in the production of reactive oxygen species during oxidative phosphorylation. So it can also result in increased mutation rate in mitochondria. Now, how does this reactive oxygen species form in mitochondria is that, you know, the electron transport chain in mitochondria is involved in ATP synthesis through creation of a proton gradient. So when the electrons move through the electron transport chain, there is a tendency that some electrons will leak out of the chain and they will interact with the oxygen, producing superoxide, which is a reactive oxygen species. Okay, so this can result in the production of reactive oxygen species, which can again increase the mutation rate in mitochondria. So 2 is also correct. Then mutations reduce ROS production since electron transport is faulty. Now I told you in normal condition itself, electron leakage is there once the electrons travel through the electron transport chain, resulting in reactive oxygen species production. Now they are telling that there is a faulty electron transport chain. So when the electron transport chain is faulty due to mutations in mitochondrial DNA, then electron transfer through the chain become less efficient, and this can lead to increased electron leakage, and it can cause higher ROS production. So mutations reduce ROS production since electron transport is faulty, no. mutations increase ROS production when electron transport is faulty is the correct statement. So they have asked for the incorrect statement, so that is the answer. Then next one, there is an enhanced rate of apoptosis in the cells with mutant mitochondria. That is correct. So mutant mitochondria, there will be more ROS production. I told you just now. This can lead to increased oxidative stress and it can enhance apoptosis rate. So 4 is also correct statement. Okay, now the next one. The epigenetic regulator Demeter, DEMETER, DMA and media, MEDEA or MEA act synergetically to repress endosperm development in the absence of double fertilization during seed development in Arabidopsis. Okay. Now what is this repress endosperm development in the absence of double fertilization? That means even though there is no double fertilization, endosperm is developing. Okay. Even though usually after double fertilization only endosperm develops. That is what you have learnt generally with the case of development of endosperm in plants. Now there are some cases where the endosperm develop even without double fertilization. That phenomenon is called autonomous endosperm development. Okay, autonomous endosperm development. So there are certain epigenetic regulators in Arabidopsis that will act synergetically and they will repress this endosperm development during the seed development. Okay, so these are those two epigenetic regulators, that is Demeter and media. Now they have asked, what does this DME function as and what does MEA function as? That is your question. So this is a research article and they have already done research with this Demeter and media, and they have found that DME is a DNA glycosylase. So you have learned that DNA glycosylase are involved in DNA repair mechanism, but here they are involved in epigenetic regulation. Okay, and the DNA glycosylase, what they do is that they remove the methyl groups from the cytosine bases, and it will help in the activation of transcription of that particular DNA. Okay, that is what DNA glycosylase as epigenetic regulator does here. So DME is a DNA glycosylase. Now MEA, it is a histone methyl transferase. So histone methyl transferase, you all know, they catalyze addition of methyl groups to specific lysine or to arginine residues in histones. And this can lead to either activation or deactivation of the gene. That is, if what we have studied is that, we all have studied is that if histone 3, if in histone 3 the lysine at position 4 is methylated, it will result in activation of the gene, whereas if it is at position 9, it will result in repression of the gene. Now they have found that MEA is a DNA histone, it is a histone methyl transferase, not DNA methyl transferase. It doesn't have anything to do with the DNA. It is involved in histone methyl transferase. Now how does these act? How does this DME and MEA act to repress endosperm development? So MEA, this is the alleles of MEA. One is maternal allele and the other is paternal allele. Now the maternal allele in MEA, it is having a methyl group, so it is not transcribed or translated. Paternal allele doesn't have a methyl group. Now what does DME do? It is a DNA glycosylase, and I told you it removes the methyl group attached to the cytosine and it will result in the gene transcription translation. So DME, it will remove the methyl group in the maternal allele. Okay. In the maternal allele, the methyl group will be removed. It will get activated, and the activated maternal allele, it will produce protein that will silence the paternal allele, and as a result of this silencing, it will prevent endosperm development during seed development in Arabidopsis. Okay, this is what is happening. So your question was not the mechanism, it was just DME is a DNA glycosylase and MIA is a histone methyl transferase. Okay. So answer is 4. Now next one. Which of the following events occur during the light reaction of photosynthesis and directly contributes to the formation of ATP? So you know light energy is absorbed by chlorophyll, then there is splitting of water into oxygen, protons, electrons, then there is the electron transport chain where the electrons are passed and it will result in the production of a proton gradient across the thylakoid membrane, and this proton gradient it will help in ATP synthesis because of this proton gradient. Okay, so that is what is taking place during light reaction of photosynthesis resulting in the formation of ATP. So as per what I have told just now, you know that it is the proton gradient across the thylakoid membrane that is involved in the formation of ATP. So your answer here is option 4. But we will see the other options also. Splitting of water into oxygen, protons, electrons, yes, that take place during the light reaction, but it doesn't contribute to the formation of ATP. They have asked directly contribute. Okay, so it doesn't contribute directly. Then transfer of electrons from photosystem 1 to NADP plus to form NADPH, that also no direct contribution to ATP synthesis. Carbon fixation by the enzyme Rubisco in the stroma. Okay, that is a dark reaction. The question is for light reaction of photosynthesis. So that has nothing to do with ATP formation, but it is involved in ATP consumption. Okay, carbon fixation is involved in ATP consumption rather than formation. So 3 you can take it as wrong answer. So what is left out? 4. Establishment of a proton gradient across the thylakoid membrane. Then, which one of the following statements with regard to glyoxylate cycle is incorrect? Okay. Now, glyoxylate cycle, it converts fatty acid into carbohydrates and it is found in plants and some microorganisms. So, it is absent in humans. That is a correct statement. They have asked for the incorrect statement. It occurs in specialized microsomes. Okay, and glyoxysomes, not microsomes. It is peroxisomes and glyoxysomes. What are microsomes? They are membrane bound vesicles formed from endoplasmic reticulum when you lyse the cell during tissue homogenization. That time only these microsomes are formed. They are not present naturally. Okay, then this cycle, so that is the incorrect statement. Okay, it is not microsomes, it is specialized peroxisomes and glyoxysomes found in plant cell. This cycle allows for the synthesis of sugars from fatty acid, that is correct. It involves enzymes of both glyoxysome and the mitochondrion. Yes, some enzymes of the citric acid cycle are utilized by this glyoxylate cycle. So, mitochondria enzyme also are used. So 4 is also correct. Then next one. In a population with the ABO blood group system, if the frequency of the allele IA is 0.3 and the frequency of the allele IB is 0.2 and the frequency of the allele small I is 0.5, what would be the expected percentage of population with blood group A, considering that the population is in Hardy-Weinberg equilibrium? Okay. So, In the ABO blood group, the blood group A is determined by the genotypes capital IA and IA and small I. So IB allele, frequency of IB allele, we need not have to take into consideration. Okay. Now frequency of the IA is IA square, that is 0.09. Frequency of IA small I is 2 into IA into I. So substitute the values, you will get 0.3. Then total frequency of the blood group is, add this, it is 0.39 and convert it into percentage, you will get it as 39%. So answer here is option 3. Okay. The disease phenylketonuria is caused by the null allele of the gene phenylalanine hydroxylase. Okay. Now what is this null allele? Null allele is nothing but it is a version of the gene or an allele of the gene that has lost its normal function. Okay. And it is an example of recessive mutation, that is, if the wild type is capital A and the mutant is small a, then a recessive mutation means it will have both small a to be present to cause the disease. Okay, the person should have the genotype small a to cause the disease. But if if it is heterozygous condition, that is capital A small a condition, then the capital A is more than enough to produce the wild type phenylalanine hydroxylase and the person will not exhibit the disease. Okay, that is what is meant by this statement. So which of the following terms best describe the wild type allele of PHA? So now you know the answer. I told you if it is capital A, then the person will naturally have no disease, that is, he will not have the phenylketonuria disease. Whereas if he is heterozygous capital A small a, then one copy of that particular capital A is sufficient to produce the normal phenylalanine hydroxylase gene protein, and the person will not have phenylketonuria. So that condition where one copy of the wild type allele is sufficient to produce the normal phenotype, it is called as haplosufficient. That is somewhat like your dominant condition in a heterozygote. So that is called haplosufficient. That is the answer. Then gain of function, that is nothing but a mutation which result in a new or enhanced function of a protein. That is gain of function. Here that is not the case. Then epistatic means one gene affects the expression of the other gene. That is also not seen here. Then allele with additive effect means each copy of the allele will contribute to the phenotype of the trait. Okay. So if capital A small a is there, some portion of it will contribute to the disease, or some portion of capital A will contribute to the disease. A portion of the small a protein will contribute to the disease. So both together they will contribute to the disease. That is not the case here. So allele with the additive effect is not correct. Next question. A mutation in Drosophila gene causes a reduction in eye size if animals are grown at 29 degrees but not at 18 degrees. That is, if you grow the animal at 29 degrees, mutation is there. If you grow it at 18 degrees, no mutation. That makes it temperature sensitive mutation. So you can cross off 1 and 3, they are incorrect. So 2 and 3 we will see. Okay, they have said whether it is recessive or dominant. So they have told in the next statement, this happens even if the animal bear a single copy of this mutation. That means it is dominant. Okay, even if single copy of the mutation is present, that is, it should be in the dominant condition, only then it will suppress the recessive gene and it will express itself. So it has to be dominant. So the answer here is option 4, temperature sensitive and dominant. So thank you for watching my channel. Please do like, share and subscribe. Thank you.